Hello, everyone. Thank you very much for joining us tonight. We are very honored to host Professor Brian Timko with the very kind sponsorship of the Society of Chemical Industry. Brian Timko is an assistant professor in the Department of Biomedical Engineering at Tufts University. He earned his PhD at Harvard University under the mentorship of Charles Lieber. During that time, he studied silicon nanowires and demonstrated the first subcellular nanoelectronic interfaces with living systems, including cultured neurons and cardiomyocytes, as well as heart and brain tissues. He then completed postdoctoral studies as an NIH NRSA fellow at MIT in the lab of Robert Langer and Daniel Kohane. Then there he studied fundamental interactions between nanowires and 3D engineered cardiac tissues, and demonstrated that nanowires could recapitulate the nanotopography of extracellular matrix, guiding cardiomyocyte assembly and tissue maturation. From there, he was an instructor at the Boston Children's Hospital and Harvard Medical School, where he studied plasmonic gold nanomaterials and their utility in near-infrared triggered drug delivery systems. Brian's laboratory at Tufts focuses on bioelectronics and is developing novel materials to achieve two-way electronic communication, as well as bioelectronic scaffolds to achieve seamless device integration with engineered tissues. Brian was recently awarded the Nano Research Young Innovators Award in nanobiotechnology and now serves as Young Star Editor for that journal. And now I'm going to hand over to Brian to introduce us to his groundbreaking research.
Thanks so much for that very kind introduction, Tina, and to Society of Chemical Industry and Chemical and Physical Society. Thank you so much for hosting me today. It's really a privilege and a pleasure to chat with you today. And I'm excited to tell you about some of my lab's recent results as they relate to bioelectronics, which we can use for disease modeling and also some prospects for making new classes of engineered tissues. So what I would like to do today is focus on two recent works that came out of our lab. The first one is a heart on a chip in which we use stable bioelectronic interfaces to understand electrophysiology in a disease model system. The second line of research relates to photocross-linkable silk fibrin, which is a new type of material that we can use in bioelectronics to further enhance stable device integration with living tissues and also to make a path forward to new classes of 3D tissues with embedded devices. So sort of the general motivation behind this work is how can we can make engineered tissues in the lab. We can design polymer scaffolds that support the growth of those tissues and their maturation into functional tissues that could be used in regenerative medicine, for example. But is it possible to stably embed bioelectronic devices inside these systems? And so these sorts of devices could provide readouts that would enable us to monitor the state of the tissue over time. They could also provide inputs, which would allow us to achieve highly localized stimulation inside the tissue, which could mimic bioelectronic inputs and also drive tissue maturation in new areas. So the scheme here that I've shown is from Charlie Lieber's lab, which is a kind of injectable bioelectronic system. But this problem really requires us to, one, design the devices, and two, to design interconnects and polymer supports that enable those devices to be stably integrated inside of the tissues. So these are some of the challenges that we're facing and focus of our research recently. So I'm going to start with our heart on a chip paper, which was published in Nano Letters not too long ago. So now the general overview here is, so we're interested in studying the effects of a myocardial infarction in which there is oxygen and nutrient blockage, which affects the cardiac function. Now, really what this comes down to is that proper tissue function is reliant upon bioelectricity. So for example, neurons fire action potentials, and those action potentials trigger neurotransmitter release, which is how signals are propagated throughout the nervous system. Similarly, cardiomyocytes fire action potentials, and these action potentials are responsible through a cascade of events for actin-myosin contraction and ultimately contraction of the cardiomyocyte. And so if we can understand how this electrophysiology works inside the cardiomyocytes and inside the cardiac tissue, then we can better understand how the tissue functions. And if we can understand how electrophysiology responds to events such as depriving the tissue of oxygen, then we can better understand how diseases affect the cardiac tissue. Now, this is not a simple problem to address. Measuring cardiac electrophysiology requires tools that aren't always easy to use. So for example, one common tool is the patch clamp electrode, which is a macro scale device that attaches to a single cell. And it can record signals with very high signal to noise resolution and very high temporal resolution. But these devices cannot be multiplexed. They ultimately destroy the cell. So they really aren't good options for studying long-term how tissues function. Another option is optical dyes, where the cell fluoresces when it fires an action potential. But these too require sophisticated instrumentation. Optical dyes are not really amenable to measuring how signals move in 3 dimensions. And again, there's a limited lifetime over how long the experiment can be performed. And so this is one example of where bioelectronic devices can be very useful. They can sit outside the cell or inside the cell, but generally we can design them to interface with these cells to record signals, bioelectronic signals, over very long periods of time. So for this first part of the research, I'm going to tell you about a bioelectronic chip that we developed that enabled us to culture cells on top of bioelectronic devices. And through a microfluidic channel, we could rapidly modulate the cell culture medium over time. And this allowed us to actually mimic the conditions present after a myocardial infarction, or at least one of them, oxygen deprivation in the medium. So before I really get into the nuts and bolts of this chip, I'd like to tell you a little more about how these bioelectronic devices function. So an action potential is an electrical spike of the membrane potential inside the cell versus outside. It's characterized by an upstroke that is created by a rapid sodium influx into the cardiomyocyte, a brief plateau region in which calcium is released within the cell, and then a repolarization region that's driven by calcium or potassium efflux from the cardiomyocyte. So these are how the ions flow when a cell fires an action potential. And one way in which we can measure these with bioelectronic devices is using a system that we call multi-electrode arrays. So essentially, in their basic form, we're starting with a silicon wafer, and we can design these devices on the wafer. They're gold pads, and these gold pads sit outside the cell. So these are extracellular devices. Now, when the cell fires an action potential, there's a flow of these sodium ions and eventually calcium and potassium ions. But the movement of these ions outside the cells creates potential fluctuations outside the cell. So it's essentially Ohm's law. You're moving ions through a resistive area. And extracellular devices will record a spike that's consistent with the action potential. So we can model this with an equivalent circuit diagram Essentially, your cell is in contact with your substrate with a thin layer of fluid between the two. So when the action potential fires, you're having your ion flows through that thin layer of fluid, and this gives us the potential fluctuations. The diagram on the top shows us what signals could look like from an extracellular device. So these are sharp spikes. They're about 10 milliseconds wide. And so these are helpful because they can tell us when a cell is firing. But there are other classes of devices enabled by recent advances in nanomaterials that enable us to record other domains of signals as well. So for example, if we fabricate on the surface of this MEA an array of nanopillars, we can actually design the system so that the nanopillar punctures through the cell membrane and accesses the cytosol. So when we do that, this is slightly invasive, but when we do that, we can actually record the action potential itself. And so this gives us signals that are similar to those recorded by a patch clamp electrode. But these devices can be readily multiplexed. And moreover, the cell membrane eventually seals up. And so they're really minimally invasive. Now, by coupling these two different types of devices, that is the extracellular devices and intracellular devices, we can record a broad range of electrophysiological signals and characterize how this tissue is functioning. So that's what I'm going to tell you about in this Heart on a Chip project. So we have this chip. It has a microfluidic channel attached to the top of the chip. Inside the channel, that's where we culture our cells. So in this case, it's a monolayer of cells. In this case, we're using HL1 cells, which are cardiac model cells. And through that microfluidic channel, we're consistently flowing our medium. So under normal conditions, we have oxygenated medium. But then to mimic the environment after a cardiac infarct, we're going to change that medium to medium that is essentially deprived of oxygen. So it's about 1% in the hypoxic medium. And this system is going to allow us to, one, rapidly modulate from normoxic to hypoxic medium, which mimics the effect, the acute effects after a cardiac infarct. But during this experiment, we can continuously read out the function of the cardiac cells. Anyway, this is what the cells look like. They form a monolayer inside this microfluidic channel. They are coupled to each other. And if we take a video of the cells, after about 3 days, they beat spontaneously. Now, significantly, they're beating spontaneously in unison, which recapitulates healthy cardiac function. So this is in the healthy state. The first question that we ask is, once we build this chip, can we actually mimic hypoxic conditions inside these cardiac cells? So remember, we're changing the medium to this 1% oxygenated medium. And so to track whether or not the cells actually become hypoxic, we can track this protein, hypoxia-inducible factor 1 alpha, HIF1 alpha. And we know that during hypoxia, this HIF1 alpha will translate into the nucleus of the cardiomyocyte, where it binds with the HIF1 beta subunit. And this initiates a cascade of protective signaling pathways that are essentially designed to prevent the cardiomyocyte from dying in response to hypoxia. So in this first set of experiments, we fluorescently labeled the HIF1 alpha, and we observed that during these hypoxic domains applied to the cardiomyocytes, we could see the HIF1 alpha actually translate into the nucleus. So HIF1 alpha in this case is labeled in red. In this case, we maintain hypoxia for 5 hours, and then we return the cells to the normoxic state. Sorry. So when we return the cells to normoxia, we saw that this complex, HIF1 alpha and HIF1 beta, dissociated, and the HIF1 alpha located outside of the nucleus. So in other words, The cardiomyocytes recovered and returned in this experiment, at least, to normal cardiac function. So this shows us that we can obtain cells that actually become hypoxic in this system. Now, the next question that we can ask is, how can we use these extracellular devices to record signaling in these cardiac cells during this process? So this is an example of a typical device. The pad itself is 30 microns in diameter. Now, the interconnect in the pad is made out of gold, but what we do is we coat the electrode with platinum black, which essentially highly increases the surface area of the device. And this reduces the impedance by about 2 orders of magnitude. So why do we want to do that? Well, if you go back to that equivalent circuit diagram that I showed you earlier, the magnitude of the signal is going to be dependent on the impedance of your device itself, because you're essentially building, if you've taken an electronics course, you may be familiar with voltage dividers, which is what we have essentially in this situation. So we can tune the surface of this device to minimize its impedance and maximize the signal to noise ratio. Now, once we do that, we can record these extracellular spikes with very high signal to noise. So this is an example of a train of beats in these cardiomyocytes. So each spike corresponds to an action potential or a contraction. Now, if we expand one of those spikes, you see a positive and negative phase. So remember, these are not the action potentials themselves, but rather the signal that's produced outside the cell as per that equivalent circuit diagram. So essentially, the upstroke of this signal is your sodium ion flux. The downstroke is caused by recharging of the capacitive cell membrane. But regardless, these data now enable us to understand the rate and periodicity with which the cardiomyocytes are contracting. So in this case of normoxic cells, you can see that the beating is regular, which is what we would expect. So now what we can do is apply a hypoxic domain to our cells and monitor over this domain how their electrophysiology changes. So here we're showing an example of a hypoxic domain. It's 5 hours long, like we showed with the GIF1 alpha experiment. And to the right, you're seeing exact representative recordings of these extracellular spikes over the course of the hypoxic domain. Now, interestingly, you can see that at the 300-minute mark in this experiment, the beat rate substantially decreases, and also the beating becomes arrhythmic. So this shows that we can, during this hypoxic domain, actually induce irregular cardiomyocyte activity. Also, we can introduce normoxic medium at the end of this experiment. And so at that point, the beating returns to normal, and the rhythmic beating is restored. So this would relate to a condition called reperfusion when oxygen is restored to the cardiomyocytes. Now, in the context of these studies, we're focusing mostly on the hypoxic domains. Now, these experiments will tell us how the cells are beating at a particular point. Okay, but that's really only half the story, because what we really care about is how signals propagate through this monolayer of cardiomyocytes. So in order to do that, we need to record these signals at multiple spatially distinct locations throughout the monolayer. And so this is where bioelectronics really shines, and especially these extracellular devices, because we can fabricate large arrays of the devices and measure signals from them simultaneously. Now, this is an example of a typical experiment where we're recording devices, recording signals from 16 devices, in this case 14 out of the 16 produced good, high quality signals. And then what we can do is apply algorithms to these signals to actually measure the time shifts between each of those different channels. So if you look closely, you'll see that the peaks are shifted in time just a little bit. And when we do that, we can construct these isochronal maps. So what do these maps show? These maps show how a wavefront is propagating through the cardiomyocyte monolayer. So each line on these isochronal maps is basically a location of the wavefront as it moves through the monolayer. Now, this shows us a couple of things. One, the monolayer is moving at consistent speed throughout the monolayer. And two, the shape is fairly uniform. So it's a uniform propagation. So in this case, we can calculate the speed. It's about 20 millimeters per second, which is more or less consistent with what we would expect from a healthy cardiomyocyte system. But when we introduce hypoxia, something very interesting happens. So you can see that these very nicely ordered isochronal maps, after just 1 hour of hypoxia, they become highly disordered. Okay, so the speed changes throughout the monolayer. Also, the direction keeps changing. So why does that happen? So during hypoxia, two things happen in cardiomyocytes. So one is connexin proteins, which are responsible for coupling between adjacent cells. Their expression is reduced. So the cells are not as well coupled to each other. Secondly, the cells actually become more excitable during hypoxia. And so that leads to multiple initiation centers of beating throughout the tissue. And so this is one reason why cardiac ischemia is so dangerous, because these cardiomyocytes begin to fire irregularly and not in sync with each other. And we can observe a constant trend over the course of our hypoxic domain, where the speed continually decreases over time. And so the gray area shows the standard deviation among a single sample, and you can see that the standard deviation also becomes much larger. Now, what's important about this measurement is that we're able to really monitor this change in real time over the course of hypoxia, which is very difficult to do using these standard techniques. Okay, so this is kind of really only half the story, because as we said before, these extracellular spikes aren't the same thing as the action potential. And so if you've learned about patch clamp electrophysiology, you know that kind of the strength of that tool is that you can really understand how different ion channels in the membrane are functioning. And the types of ion channels that are functional are also responsible for cardiac function. So this is where nanoscale devices can really come into play. So I'm going to show you another structure that we studied, these platinum nanopillars. So we can take these extracellular electrodes that we've been studying, and instead of coating them with platinum black, we can fabricate on the surface these nanopillars that stick up from the surface of the electrode. Now, these are about 100 nanometers wide, and they're about 2 microns tall. So when we culture our cardiac monolayer on top of these devices, the cell membrane will conform to wrap around the pillars. But we can do something interesting here. We can apply an AC electric field to these electrodes, and the field is enhanced at the tip of the nanopillars, which causes the cell membrane to locally electroporate. So we can poke holes in the cell membrane. And when that happens, these nanopillars can actually access the cytosol. Now we can observe that happening within our heart on a chip device. So what you're looking at here on the left is the extracellular spikes that are recorded by these nanopillar devices. So you'll see the signal to noise here is a lot lower than what we've recorded before because the impedance of these devices is much larger. So remember, they're very small. But what happens is when we electroporate the membrane, we see an immediate increase in the magnitude of the signals that's recorded. And if we expand them, as you see down below, these have the same shape as the actual action potential. So in other words, when we poke these nanopillars into the cell membrane, into the cytosol, we can actually record action potential like signals. Now, over time, the cell is going to reseal and expel the nanopillar from the cytosol. Now you can see that from the signals recorded up above, where their magnitude decreases over time, and eventually they transition back to the extracellular signals. So this is actually a very good thing because it means that we're not killing the cardiomyocytes, which means that we can probe them multiple times over our time domain. And whereas the amplitude of these signals is not going to be quantitative because it's changing, if we normalize the signals that we're recording, we see that the shape changes the same. So we can study how the shape of these signals changes over time in response to a hypoxia to understand how cardiac function changes. So this is what we did. The trace at the top shows what these action potentials, so the intracellular signals, look like at the end of 6 hours. So similar to the extracellular signals, you can see that these become arrhythmic and sparse. But we can also monitor what's happening more or less continuously over the time of the hypoxic domain. And so here we see at 0, 2, 4, and 6 hour time points, the width of the action potential decreases by about 50%. Now, this is consistent with some studies that look at potassium ion channels in the cell membrane. So there's a class of potassium ion channels that are ATP dependent, and their function actually increases when ATP levels decrease. So this increased activity of the potassium ion channels in response to hypoxia is going to make repolarization of the cell membrane faster. So that reduces the width of the action potential. And so this is really one of the first times where we've been able to observe this phenomenon in real time. So this is pretty interesting because it lets us look at how these action potentials change in response to hypoxia. But you might be saying, okay, well, you can kind of do that with patch clamp electrodes if you get a good patch or if you patch multiple cells over time. But same as with extracellular devices, these intracellular probes can also be multiplexed. So to demonstrate this phenomenon, we looked at arrays of 5 spatially distinct intracellular probes. And so we could record these multiplexed action potentials under either normoxia or hypoxia. So you can see representative signals here. But what we can actually do is measure the width of these signals. And we can see that in this system, the width of the signals in these 5 different locations also decreases. So we're representing those data through these heat maps. Now, the reason this is significant is for a couple reasons, actually. So one, cardiomyocytes are not homogeneous. Okay, they're heterogeneous in their function. So it makes sense to be able to probe multiple cells at the same time to understand how their function changes in response to hypoxia. Now, secondly, in this system, we're applying the same extent of hypoxia to all the cardiomyocytes. But endogenously, you would experience gradients of hypoxia. And so sort of the next question that we can ask is, how can we map cardiomyocyte function in response to hypoxic gradients? So this is one outstanding question that we're working on in our next steps. But these data really kind of drive home the point that we can actually record signals that allow us to understand ion channel function throughout a cardiac monolayer and to map how those signals change. Okay, so these studies have all been confined to cardiomyocyte monolayers. Now, monolayers of cells do not really recapitulate 3D tissue function. And so it really becomes necessary to push your studies into 3D domains in order to make a really clinically relevant system. And so this requires us to design new classes of bioelectronic devices that can actually be embedded within the tissue. So this brings me to the second part of my talk, where I'm going to tell you about a new type of material that we designed that sort of incorporates some properties of silk, which is a common material that's used in tissue engineering, and sort of translates it into the domain of bioelectronics. So in other words, we can design scaffold-like devices that also incorporate bioelectronics. Now, before I tell you too much about silk, I'm going to take a step back and just show you some kind of more conventional devices that we have designed in the lab. Now, if you take those recording elements that we discussed in the first part of the talk, you can fabricate them onto flexible polymer supports rather than silicon wafers. So one common material is called SU8, which is essentially a photocross-linkable epoxy. So it's compatible with clean room processes, but we can really, the support is entirely polymer based. Now, this is one example of a type of device that we fabricated. So you can see that it's flexible, it's clear except for where the gold is. And most significantly, if you look at the expansions here, the electronics itself is actually a mesh-like geometry. So this device is mostly air. So if you were to embed it within a 3D tissue, in principle, you would not perturb cell-cell coupling around the device. Okay, so this would enable us to build truly embeddable devices. So this is the recording elements side. We can also fabricate stimulation elements on the same kind of SU8 material. I'm not going to get into stimulation too much in this talk, but long story short, we can achieve highly localized cellular activation using these different types of stimulation elements. Okay, so this is SU8. It's a common material, and it is bioinert, which means that it doesn't really, it's not really toxic to cells, but it's not bioactive either, which means that cells don't really like to stick to it very well. And even if we coat these devices with adhesion factors, cells still don't like to stick to them very well. And so this is a problem in bioelectronics, because to record high signal-to-noise ratio signals, we really need very good interfaces between the cells and the devices. So we took some cues from tissue engineering, and we asked ourselves, okay, can we take silk, which is used in cardiac tissue engineering, and can we modify it so that it becomes photoactive, so that we can incorporate it into conventional photolithography processes to make bioelectronic devices where the SU8 is substituted with silk. And so what we did was we took silk fibroin, which is a component in silk, which this is the component that's usually used to make like hydrogel tissue scaffolds. We methacrylated the backbone. And so methacrylate groups, in the presence of a photoinitiator, will cross-link with each other. So what we could actually do is make these methacrylated, create a suspension of this methacrylated silk, and we made essentially a new kind of photoresist. So we could spin coat this material onto the surface of a wafer, selectively expose with UV light. So we did this in the clean room, same as when we fabricated our planar devices. But we could actually selectively cross-link this material and then develop it, same as we would for other types of photoresist materials. So we call this photocross-linkable silk fibrin, and it exhibits some very interesting properties. So first of all, we can obtain centimeter scale uniformity in the patterns that we fabricate. We can also get very small feature sizes, so down to less than a micron minimum feature size, which is much smaller than the resolution threshold that we would need to fabricate bioelectronic devices. Also significant, we can control the thickness of these films by modulating the concentration of PSF in the photoresist or the spin speed. And also, if we fabricate all of these structures on an underlying sacrificial scaffold that's subsequently dissolved, we can actually lift these structures off the surface of the substrate to achieve freestanding silk structures. Okay, so this demonstrates that we can achieve these silk structures in whatever kind of pattern we want. But the next question that we could ask is, are these things actually suitable for bioelectronics? So in order to answer that question, we need to, one, look at the electrical properties of the material, and two, look at how cells actually adhere to the material. So with response to electrical properties, essentially, this photocross-linkable silk scaffold needs to support the devices, but it also needs to passivate the electrical interconnects. So essentially the wires that connect the devices to the outside world, they need to be encapsulated within the silk, and the silk needs to be insulating. SU8 is insulating, and it also is very robust. It can last for many months in cell culture medium. But we really had no idea whether this photocross-linkable silk would behave the same way. So we performed a series of impedance spectroscopy measurements on these monolayers. And essentially what we're doing is we're taking a monolayer, or not a monolayer, but a thin film of silk fibroin, putting an electrode, we put an electrode on either end, and then we apply an AC signal and look at how much current is passed through the film. So this allows us to understand the electrical properties of the material. And so interestingly, this photocross-linkable silk fibroin functions similarly as silicon dioxide, which is commonly used as a passivating layer in bioelectronics. So this was very, very encouraging to us. Now, to further test this point, we fabricated an array of MEAs, same as we did in the heart on a chip model. But in this case, instead of passivating the interconnects with SU8, we passivated them with our photocross-linkable silk fibroin. And then these cells contracted, and we measured the extracellular signals, same as we did before. And so this is pretty exciting. We were able to measure these signals over 8 days, and you can see that the cells increase in magnitude, the signals increase in magnitude, which we would expect because the cells are becoming more electrophysiologically active over the course of the experiment. But also the signal to noise ratio is very high. So this sort of also goes with the previous experiment to tell us that these silk films are doing a really good job of passivating the interconnects to the extent that we can perform bioelectronic studies. Okay, so this tells us something about the electrical properties of the material. The final question that we can ask is, do cells like to adhere to this photocross-linkable silk fibrin? So to perform these studies, we switched back to silicon wafers, and we fabricated a crossbar motif of photocross-linkable silk fibrin on the surface of the wafer. So you can see schematically what it looks like here. And we coated everything with polylysine, which is a common adhesion factor. So we then adhere to these surfaces neurons. So these are neurons that were derived from a cell line called human-induced neural stem cells. So what happens is the cell line spontaneously differentiates into human-like neurons after a couple of days. And in this experiment, we then stain the neurons for beta-3 tubulin, which stains for a neuron-like lineage, and DAPI, which stains the nucleus. And so this allowed us to image the neurons on these structures. So here we're directly comparing on the right, SU8, which is the conventional material, on the left with photocross-linkable silk fibrin. And so these are after 8 days of culture in vivo and in vitro. And so what we can see is that the neurons on the SU8, over time, they actually migrate off of the SU8 structures. But the neurons that are cultured on top of the photocross-linkable silk fibrin, they do not migrate off. So this is encouraging for bioelectronics because it means that we can actually achieve kind of good adhesion between these structures and between the neurons. So we started asking some questions about why this could be, why our PSF performed so much better than the SU8. We performed some contact angle measurements. So essentially the contact angle tells you about the wettability of the substrate. And so we can start with SU8, and we can see that after coating the SU8 with polylysine, the contact angle doesn't really change at all. So in other words, SU8 starts more or less hydrophobic. It stays hydrophobic after we coat with SU8. Photocross-linkable silk fibrin, on the other hand, is much more hydrophilic, and it remains hydrophilic after we coat with polylysine. And interestingly, if we use a type of polylysine that is conjugated to a fluorescent dye, we see in the case of SU8, there's really no polylysine on this crossbar motif. But if we move to the case of PSF, you can see that the polylysine adheres very well. And so we really want polylysine to very well coat these devices, because that's in some cases, that's crucial for cellular attachment and adhesion. So this really drives home the point that these photocross-linkable silk materials could be a promising route for bioelectronic studies. And so I think I had 45 minutes to chat with you, and I think I'm about at the 45-minute mark. So what I'm going to do is wrap things up. So basically, we've developed this heart-on-a-chip model. We've shown how it can be applied to one kind of disease model. And then we started to look at this new photocross-linkable silk fibrin material that could enable us to translate these devices into the 3D tissue constructs. So our studies in this area are continuing. One, to look at the electrical properties of 3D engineered cardiac tissues and brain tissues, but also to incorporate stimulation capabilities, which could give us these two-way electronic communication with the cells. So I'll wrap things up. Tufts is a great place. If you have the opportunity, I would highly encourage you to visit. And of course, none of this work would have been possible without my students and our collaborators. So I will leave things there. Again, thank you for giving me the opportunity to chat. And I guess if there are any questions, I'd be happy to answer them now.
Thank you very much for the fascinating talk. It was really impressive to learn about bioelectronics and the new advancements in the sphere and how it can connect to heart tissue and even neurons. So now you can ask your questions. If anyone has a question, you can type in the chat or raise your hand and unmute yourselves.
Can I start with the first question?
Yeah, of course.
Thank you so much, Brian, for a fantastic talk.
Absolutely.
Okay, Kara, you can go and then we can go from the lecture theater, I guess.
Thanks.
That's okay.
I'm sorry, my question isn't very scientific, but I wanted to ask, how did you get to where you are, and were there any moments in your career that you really felt were defining moments for you?
Yeah, that's a good question. And so when I started my PhD program, I really didn't work with biology at all. And sort of at the time we were investigating how nanomaterials could be used in computation, sort of to make nanoscale logic devices or nanoscale memory structures. And sort of it was a gradual progression. At one point, we started using nanowires to build chemical sensors, and sort of this was at the point where I started to incorporate these devices with at the time neurons, and so that was really the first time I ever did cell culture. So those experiments worked, and so that sort of started my general progression from kind of being this solid state person who made silicon structures to someone who started working with cell culture. And then sort of during the postdoc, I experienced some more solid biomedical engineering applications with doing sort of more types of tissue engineering and then eventually drug delivery. So I mean, I guess the answer to your question is I started out expecting to do one thing, and then I wound up doing something totally different. So I guess the key here is sort of be open-minded in science and just kind of pursue avenues as they open up.
Thank you so much. I really enjoyed the talk.
I believe we have one question from the audience.
Yep, of course.
Hello, thank you very much for the talk. And obviously from the talk, you can see that the silk scaffolds are very well for incorporating into tissue structures. If you were to introduce it into, say, a larger in vivo system, what do you think the immune response would be?
Yeah, so that's an excellent question. And so, so generally, immune responses in vivo are driven by well, they could be driven by chemical factors, but with things like SU8 implants or silicon implants, they're not really cytotoxic, but there are mechanical mismatches between the tissue and the device because really the devices aren't squishy. And so if you take a conventional implant and you say you put it in the brain, over time, a fibrotic capsule will form around the device, which will inhibit the fidelity of device recordings. Now, we haven't looked at photocross-linkable silk in vivo, but what I can say is if you look at the case of SU8, that material is still pretty stiff, but we can fabricate it in structures, nonlinear structures like serpentine geometries that sort of uncoil, which kind of impart stretchability. And in fact, SU8 meshes have been injected in vivo in a rat model in Charlie Lieber's lab for up to 9 months. And they seem to show minimal immune response in that case, just because of the geometry of the structure. So, I mean, for the silk, we'd have to look at that for sure. I imagine it would probably be amenable since silk is used in tissue scaffolds, but it's kind of an open-ended question. I think sort of the other half of that question is how does the material degrade over time? How does that affect the fidelity of the signals that we might measure? We know that photocross-linkable silk is robust over at least 3 weeks. We haven't really looked at further time points, so that's an open-ended question right now as well.
Thank you so much for answering the question.
Thank you. Someone in the chat had a similar question if the materials can lead to allergic reactions, but I think that the answer covered that. Does anyone have another question? In the meantime, I can ask a question. I've heard, I think I've heard somewhere that sort of smaller infarctions can be sort of protective against larger infarctions. So I was wondering if that can be tested with bioelectronic methods.
Yeah, that's a really interesting question, and I'll have to admit I'm really not I don't really understand enough about the physiology to understand for sure how the mechanisms behind that. But I think one advantage of these devices is that you do get very high spatial resolution. The intracellular probes are really monitoring a single cell. And the extracellular probes are maybe not necessarily a single cell, but probably several cells. So really the question comes down to how dense can we make these device arrays? And so in this case, I showed 16 devices. We have gone up to 32 devices. In principle, these 30-micron pads could be shrunk down to a couple microns through some innovative kind of creative coating techniques. So that would reduce the footprint of the device. And in principle, you can increase the density of these devices substantially. So I've seen at least 1,024 device arrays. Now they require some kind of sophisticated electronics because you've got to address all of them and deal with all of the data. But definitely that is possible as well. So I guess the answer to your question is if you wanted to induce a very highly localized infarct-like condition with the right kind of device design, I think you would be able to derive some useful information from that, yes.
Thank you very much for answering. Do we have another question from the audience? You can type in the chat if you don't want to unmute yourselves. We have a question from the lecture theater because I can't see from the audience there. couldn't hear.
I believe we, and this also does.
Sorry, I couldn't hear.
I believe we don't have any more questions from the audience.
We have a question in the chat that just came in. Do you consider that personal medicine is significant? And can the bioelectronics be used as personal sensors?
Yeah, I mean, I think that's a really great question. And sort of one outstanding challenge in the field is to say, can we record electrophysiological data, sort of in real time, analyze that data, and then use that data to tailor the kind of therapy. So for example, could we take readouts from the heart and then sort of use data reduction and classifying techniques to understand, okay, is this healthy cardiac function, or is it unhealthy function, or is there something small about the data that indicates that the heart is gravitating toward unhealthy function? And then could we supply, say, localized pacemaker pulses to correct the deficiency? And so, so those sorts of challenges require us to design the devices, also computer algorithms. And this is where things like machine learning is very powerful now. And so it's kind of a lot of really new and interesting opportunities there, I think.
Amazing. Thank you very much. Thank you. Yeah, it is very difficult. Thank you. From the chat, I'm reading out them.
It's a big challenge now, for sure.
I think if we don't have more questions, I can stop the recording. But before that, I want to say thank you again for the amazing talk. It was very interesting to hear about that science. Sorry. Yeah, a round of applause. I'm sure the people in the lecture theater are clapping now, but because of the noise, we can't really hear it over Zoom. And yeah, before that, I also like to thank you for the talk. And I would like to thank to the SCI for their sponsorship throughout the pandemic on behalf of the CPS committee. And I would want to say that students can become a member of the SCI for £25 per year. And student memberships allow the opportunities to develop your network and career. And also you can go to conferences at a discounted rate. And you get a subscription for the Chemistry and Industry magazine. And there are members of the SCI present in person in the lecture theater, so you can ask them questions on how to get involved in the SCI. And again, thank you very much to everyone who came, and stop the recording.